So, Lecture 1, The Evidence for Evolution. You almost certainly won't recognise this gentleman here. This is Theodosius Grigorovich Dobansky, which I'm sure I've pronounced very badly indeed. Dobansky, however, said probably one of the wisest things ever said, and it was this: nothing in biology makes sense except in the light of evolution. Now, this is true. Evolution is the unifying theory within biology. It's the one fact that makes sense of pretty well all the other observations. Dobansky was an interesting character for a couple of reasons. One of them was that, together with others, he was responsible for bringing together Darwin's ideas on natural selection and sexual selection, together with Mendel's findings on genetics. And he brought those together in what's called the modern synthesis, which is just as well because towards the end of the 19th century, Darwin's ideas were beginning to fall a little bit out of favour. Dobansky's also interesting because he was a theist. He believed in God. He was an Orthodox Christian. And I just want to make one point, really, before moving on to the meat of this lecture. I'm aware that for some, there's a perceived and irreconcilable mismatch between theistic belief and evolutionary biology, not least, of course, because there are some very vociferous evolutionary biologists tilting at religion. I think all I want to say really is that there are very eminent evolutionary biologists who are also theists. A couple here for you. The one on the left, Francis Collins, headed up the Human Genome Project, the first time the human genome was sequenced back about 20 years ago. And the chap on the right is Simon Conway Morris, who is a professor of evolutionary paleobiology at Cambridge. Both pretty good at their jobs. Right, so 5 bits of evidence then. How do we know evolution is true? 5 ways, really. I'll just quickly introduce these. One is the fossil record, most notably because there are transitional fossils between major groups of animals. So perhaps the best known one is the evolution of 4-legged tetrapods from fishes. Another one, for example, might be the evolution of birds from dinosaurs. And fossils allow us to see the gradual acquisition of characters in that process. Secondly, homology. Homology is a special kind of similarity, really. Structures are homologous if they are similar because they share something that was present in the common ancestor. So your hand and the hand of a chimpanzee are similar because your common ancestor with a chimpanzee also had a hand like that. But we can trace it back much further, and tetrapods, all living tetrapods, have a basically pentadactyl limb, a limb with 5 digits. And that's because we can trace that all the way back through evolution to a common ancestor which had 5 digits. There's not only homology in terms of anatomy, that's to say morphology, but there's much deeper homology in terms of genes and molecules, and we'll look at a little bit of that. Thirdly, there are vestigial structures. These are things which are sort of left behind in the process of evolution. Perhaps the best known is your appendix, which, so the story goes, doesn't really have a function, but you've got one because it's kind of a leftover. We'll look at that in a little bit. Fourthly, biogeography. It's a fairly obvious statement that not all species are found everywhere, but it's really thinking about the distribution of species across the globe that led Darwin into thinking about evolution, and also led his colleague Alfred Russel Wallace thinking along similar lines. And finally, and fifthly, direct observation. We can actually see evolution happening around us. We can certainly see selection, natural selection and artificial selection taking place, but we can also occasionally see the process of speciation underway. So firstly then, the fossil record. Perhaps, as I say, the best known transition, big evolutionary transition, and it's an iconic one, and it's captured in this Darwin fish here, is the transition between fishes on the one hand and four-legged tetrapods that are able to walk around on the land, like us, for example. Now, that transition from things which are unquestionably fishes, these sarcopterygian fishes at the bottom, lobe-finned fishes, and things which are unquestionably tetrapods, they've got four legs and they're able to walk around with those four limbs, was for a long time a rather poorly known one. And in fact, the gap, the hole, if you like, in the fossil record even had a name. It was called Romer's Gap. It's about 25 or more million years long, and there were hardly any fossils from this interval. Over the last 50 years or so, it's gradually begun to be populated, and we've understood more and more about the forms which lay in between fishes and tetrapods. This is one of them. It's an animal called Tiktaalik. We don't know much about its tail. That's not been terribly well preserved, but it has a very fish-like head, but it has more so than the ancestral sarcopterygian fishes, it has more limb-like fins. It's able now to raise itself up off of the substrate, and it's acquired several characters in the skull which begin to look a bit more like the first tetrapods. Here's another animal, Acanthostega, a little bit further up the tree. You can see that again it's got a very fish-like flattened skull, but you can also see that it's got limbs. Those limbs are a bit odd in that it's got more than 5 digits. Many early tetrapods are polydactylous. They've got lots of digits. It's got a very fish-like tail still, so it's a combination of tetrapod characters and rather fish-like characters. Notice also that the ribs are rather short, they just stick out. Now, most things that spend time on the land, their ribs are very long and they curve around the body, and that provides support for the internal organs. So Acanthostega didn't have a lot of support for the internal organs, and its limbs stick out to the side. So it was probably capable of doing a quick stint on the land, maybe across a mud bank or something like that. But we think that the limbs were really used more for clawing its way through congested, weed-choked, shallow waters, rather than necessarily spending a lot of time on land. So the limbs then in Acanthostega were probably more for manoeuvring about than for walking around as we classically think about it. Ichthyostega, a little bit higher up the tree. Again, this very fish-like tail. The limb girdles, the bone structures, the bone braces that join the limbs onto the rest of the skeleton and onto the spine are better developed. The limbs are moving a little bit more underneath the body in Ichthyostega now. And the ribs, you can see, are much better developed, much thicker, and come down over and around the internal organs to provide better support. So Ichthyostega probably could hack it on the land a little bit better than Acanthostega. So that's one transition, but there are lots of transitions within the fossil record that are really rather well documented. Here's another one a little bit closer to home. This is the origin of mammals from a group within a group called the synapsids. Now the synapsids first appear way back in the fossil record at around about the same time as the first dinosaurs, back in the Permian, a bit further back into the Carboniferous. This animal here is from the Permian. It's an animal called Dimetrodon. It's within a group of synapsids called the pelycosaurs, and here you can see it within this phylogeny over on the right. You can see this enormous sail on the back, and this was heavily, had a skin membrane, we know this, and that skin membrane had lots of blood vessels in it, heavily vascularized. And we think that this was an adaptation to allow this animal to warm up a little bit quicker in the morning. And by raising your body temperature, you can become active more quickly. This is a poikilotherm, cold-blooded animal, and it relies upon external sources of heat in order to regulate its body. So it's a bit like a solar panel, allowing it to get going earlier in the morning. Of course, it's a carnivore. You can see that from its teeth, rather sharp. You can also see that its teeth are a little bit different in shape. Most lizards and reptiles have teeth that are all pretty much the same. But if you think about our teeth as mammals, we've got incisors, canines, molars and so on, and they do slightly different jobs. And these early synapsids, on the road to becoming mammals, were already starting to show differentiation of the teeth. You can also see that it has a sort of semi-erect gait. It's still a bit sprawling, but the limbs are beginning to come underneath the body. And of course, in modern mammals, or crown group mammals, the limbs are directly underneath the body, which is a much more efficient way of moving around. But anyway, this diagram on the right shows you that it's not just pelycosaurs, it's not just Dimetrodon. There's a whole host of mammal-like reptiles, as they're often called, synapsids, on the road to what we call crown group mammals, mammals that have all of the characteristics that we would list in a living mammal, let's say. That's a transition that takes something like 100 million years, and we have pretty well all of those steps documented in the fossil record. This is a rather baroque tree. There are lots of side branches that go off, and there are groups of synapsids that become enormously successful and then die out, and then other groups come along and flourish and then die out. And there's one lineage that makes it through to modern mammals. But that's, again, a remarkably well documented transition in the fossil record. Just quickly speak to this tree here. This is a tree of mammal groups, and I want to use it to illustrate the origins of cetaceans. These are whales, porpoises and dolphins and so on. I'm just going to pull out this part of the tree and magnify it. So there are the cetaceans up here, and there's a good deal of molecular and morphological evidence supporting this, but the closest living relatives of cetaceans are the hippopotamuses. Now, hippos don't look very much like cetaceans or whales, so one misunderstanding that we have to clear up is that this diagram doesn't mean that whales evolved from hippopotamuses. What this diagram means is that both hippos and cetaceans evolved from a common ancestor way back here. The other thing you can see in this diagram is lots of these branches here have got a little cross by them. So these are genera which are now extinct, so we only know them from fossils, but they document transitional forms becoming gradually more and more adapted to an aquatic or marine way of life. So I just pick a couple of the fossils from in here. I'll pick out Ambulocetus pachycetus and Basilosaurus. Ambulocetus, you can see, has a rather whale-like skull. It's very long. All of the teeth are quite similar, which is typical of the toothed whales and typical of animals that are living on fish, to have lots of quite similar pointy teeth. The spine flexes up and down, and of course whales flap their, and porpoises flap their tail up and down, whereas of course fishes and sharks and marine lizards generally flap the tail from side to side. But Ambulocetus was probably able to walk on land, probably not terribly well, but it was adapted for swimming and adapted for fishing rather nicely. This is another transitional form, Pachycetus, again a combination of aquatic marine adaptations, but retaining the ability to walk around on the land. So this is a sort of halfway house. And then this is going the whole hog, or the whole whale. This is Basilosaurus, used to be known as Zeuglodon, but it's a proper a proper cetacean. It has an enormously long tail, however, rather whip-like, and a very whale-like skull, and the nostrils migrated back towards the top of the skull, so it's got a proper blowhole now, and proper flippers. It wouldn't have been able to walk around on land at all. So here's another more close to home transition. If we go back to about 1850, so Darwin's day, we knew that humans and chimpanzees, Pan over here, were very similar morphologically. We looked very similar. And of course, Darwin talked a great deal about the evolution of humans from within the apes, or within the other apes, I should say. But at that time, all we had was chimps and humans. We didn't have any well-documented intermediate forms or intermediate fossils. This axis here shows the capacity of the cranium, the brain size, if you like. So Pan has about 400 centimeters cubed. Humans have maybe somewhere in the region of 1500. This is a big gap. If we go to 1900, wind on 50 years, then there are fossils then known of Homo erectus. So this goes back best part of 2 million years. And Homo erectus has a cranial capacity somewhere between about 750 and 1200 cubic centimeters. So it bridges part of this gap. And we also learned about Neanderthals, Homo neanderthalensis or Homo sapiens neanderthalensis, depending on who you ask. And some Neanderthals have a cranial capacity even larger than modern humans, although it's different regions of the brain which are enlarged. Wind on another 50 years and we have Australopithecines being discovered. So these have a fairly low cranial capacity, but this takes the fossil record back about 3 million years. Go on a bit further, 20 years ago or so, and the picture's much the same as the picture we have today. We've got the Australopithecines, but more groups of those, and increasing cranial capacity in some of them. We've got Homo habilis, Homo ergaster, bridging not only the gap through time, but also bridging the morphological gap. And highlighting cranial capacity here is all very well, but there are other features in terms of the construction of the skull, the construction of the skeleton, the ability to walk upright, and so on, which we can see transitioning through from Australopithecines back about 4 million years ago to modern humans. So this is more or less the picture we have of human evolution. There's modern humans there, and fossils of various lineages or various genera and species of humans. These blue lines show where fossils are known. So, for example, Homo erectus, we know its range. These white sections are where we think there's a gap in the fossil record. So the number of gaps is actually relatively small relative to the length of stuff that we actually know of. And this all joins together into a reasonably convincing tree. I just want to give you an example now of a living animal which, were it to be known as a fossil, would probably raise eyebrows. And indeed, when it was first described and when it was first stuffed, skins of this thing were first brought to the Royal Society, people thought they were a hoax. And the animal in question is the duck-billed platypus. Now, many people think that the duck-billed platypus is a marsupial, but it's not a marsupial. It's a member of another group called the monotremes. Now, just to give you a little bit of background, you probably know that there are two big groups of mammals. There are the placental mammals, and we give birth to relatively large young. We gestate for a long period, and so that includes things like us and cats and dogs and sheep and so on. And then there are the marsupials. So the marsupials give birth to relatively small young, and they make their way to a pouch, and they attach to a nipple there, and they grow, but they live within the pouch for a long, long time. So their rate of development is actually a good bit slower. So these include things like kangaroos, wallabies, and so on. Now, the divergence between the marsupial line and the placentals is way back in the early part of the Cretaceous. So this is the time when there are T-Rexes and dinosaurs crashing around. So the lineages go way, way back, and these lineages, the marsupials and the placentals, were diverging and evolving on different continents. So it's a little bit like two independent runs of an evolutionary experiment to give us mammals, and we'll come back to that a little bit later in the lecture. But monotremes are a very much older divergent line of mammal evolution. We sometimes distinguish between what we call the crown group and the stem group. And crown group mammals are defined such that they include all of the living forms and one or two fossils that get brought along for the ride. So because monotremes are still alive, because platypuses and echidnas are still about, we define crown group mammals to include the monotremes, the placentals, and the marsupials. But notice there are this great big part of the tree here where everything has now gone extinct. Now, monotremes, very, very odd. You probably know that they don't give birth to live young, but rather they lay a shelled egg. So not all mammals, therefore, give birth to live young. They also have some really weird features. So they have this duck bill in the duck-billed platypus, and it has an amazing ability to sense electrical changes within the water. So they can actually hunt even if they can't see or hear. They can hunt with this electrical sense. And the males in the breeding season, have spurs on their limbs which have a poison sac or poison gland attached to the base of it. So they're venomous. These are venomous, egg-laying, electro-sensing mammals. The females don't have nipples, so they sort of ooze milk from the mammary glands. Very, very odd, really strange combination of characters. Now, if I had presented you a duck-billed platypus as a fossil and said this is what this mammal does, it lays eggs and it has, not to mention the webbed feet, of course, but it's hairy, it lays eggs and it's electro-sensing, you'd have said, well, yeah, maybe, or maybe you're misinterpreting this, maybe you've got the wrong end of the stick here. So it's a wonderful transitional form, but it's not a fossil. It's what some people sometimes will refer to as a living fossil. And it's quite possible, quite on the cards, that monotremes may not have survived to the present day. And then we wouldn't have this beautiful example of a transitional form, but one that's actually still alive and you can still see. Another bit of evidence that comes from the fossil record is what's called superposition. So we find layers of rock, one on top of the other, that are progressively young as they're laid down. So the very old rocks are at the bottom and they tend to be exposed less often, and the young rocks accumulate on top of the old rocks. Now, this is a lovely map. In fact, it's the first ever geological map that was ever made, and it's of the United Kingdom. We're very fortunate in the British Isles that we have a hugely varied geology, and it's perhaps because of that the science of stratigraphy and particularly paleontology really blossomed here. But broadly, as you go westward in the UK, you start looking at progressively older and older rocks. And you may know that some of the first animal fossils are from rocks that are in the Cambrian period. And the Cambrian literally means Wales, because many of the Welsh rocks are Cambrian in age. Now, the person that discovered this, often called the father of British geology, is William Smith. And if you go to 26 Great Pulteney Street, you'll see this plaque on the wall to William Smith. He dug ditches and canals and was an engineer. But he noticed as he was digging down through the rocks that he encountered particular fossils, but he encountered them in a particular order, and this order always seemed to be the same. And he realized that the animals and plants he was finding changed as he went back in time. And he realized that he could correlate the rocks according to the fossils he was finding and begin to work out the order of superposition of these different rock layers. Now, this is a rather splendid rabbit, and that's the one on the right. The one on the left is a fellow called J.B.S. Haldane, was another one of these people responsible for the modern synthesis, together with the likes of Dobanski. But Haldane, another very interesting character, has a lovely quote, and he said, I don't know where it's up on this slide, there we are, he said, "I will give up my belief in evolution if someone finds a fossil rabbit in the Cambrian." So back in these very, very old rocks. And the reason for that is very simply that on the whole, we find this progression of forms from what are on the whole quite simple animals like jellyfishes and sponges, a bit later worms and so on. Those are the first animals we find, and they're also the animals which we believe evolved first. They're the ones that branch off closest to the base of the evolutionary tree. And rabbits are something that evolve much, much higher on the tree within very derived, very late evolving, relatively late evolving mammals. So that's something we expect to find in the fossil record much later. So what this alludes to is an agreement between the order in which things evolve on our best estimates of the evolutionary tree and the order in which we find things occurring through the fossil record. So the two tend to agree rather well, and that's another bit of circumstantial evidence for the process of evolution. And people will quite often plot evolutionary trees against what we know of the fossil record. So this particular tree is for a group of sea scorpions, eurypterids, and these black lines show where they're found from the fossil record. This is going back in time as we move to the left, and this has been superimposed on an evolutionary tree. And broadly, the two marry up, and that gives us faith in both, really. So number two then, we've dealt with the fossil record. Now let's consider homology. I've already given this example, but your limb, your forearm is made up of a humerus, radius and ulna, wrist bones and digits. And so is the forearm of a cat, a whale and a bat. Now, all three animals have modified this pentadactyl limb in different ways, and their mode of locomotion is rather different. We don't walk on our arms, but cats do. We've freed up the forelimbs to do other things. The whale has modified the forearm as a flipper for swimming, and the bat's modified it with a huge membrane, a huge patagium for flying. But the underlying structure, the underlying architecture of these four limbs is the same. And they are homologous because the common ancestor of all of these animals also had a pentadactyl limb with the same ground plan. And so that's the idea. We can trace back, well, here it's a whale, a human, a lizard and a bird. Different modifications, but present in that common ancestor. The teeth. Okay, here we've got a beaver on the left and an elephant on the right. So again, the way in which the incisors of the beaver and the incisors of the elephant have been modified are very different, but they are homologous as incisors because the common ancestor had the incisors too. It's simply that they've developed those structures in very different ways. Now, not only can you have morphological things or anatomical things which are homologous, but there's also an underlying very deep homology in the genetic code. Now, I'm sure if you don't know, you'll have someone telling you very soon that amino acids that make up proteins, make up polypeptides, are coded for by this triplet code within genes. So there are three bases that go together in a code to pull in a particular amino acid. And this code is semi-redundant in the third letter. So what do I mean by that? Well, let's have a look at, for example, what happens if your first base is a G, your second base is a G, so your GG, and what's the third base? Well, it doesn't matter. Whatever third base you have, whether you have a U, a C, an A, or a G, you still code for glycine. Well, okay, that's all very well, but the wonderful thing about this code is that it's universal as far as we can tell. All living things use either precisely this code or something very similar. And what that means is that we can take genes out of one organism, and transgenic organisms are those that have had genes from another organism put into them, and they're able to express those genes despite maybe 1 billion years of intervening evolution. So here's a very simple example. This is a zebrafish in the middle, which is of course a vertebrate, and there are a couple of cnidarians. So one is a jellyfish over here, the other is a coral, that produce bioluminescence. They are able to produce light, they glow. You can take the genes responsible for this from the jellyfish, and you can, using genetic engineering techniques, you can put that within the fish, and the fish is able to read these genes, or this gene, and is able to glow in the same way as the jellyfish, or in the same way as the coral, to give you a red fish. Now, you can buy these in America. These glowfish, unfortunately, it's a great pity you can't buy them here. But the divergence between jellyfish and vertebrates or fishes is at least 600 million years. So you've got two lots of 600 million years evolving independently. That's 1.2 billion years worth of evolution, and yet the fish can still read the jellyfish gene, and that's really quite astonishing. So the ubiquity of this genetic code is one of the most compelling bits of evidence for evolution. There are also regulatory genes, there's sort of master control genes that we find very widely distributed. So these are Hox genes that enable, in this case, a fly to pattern its segmented body and to tell the developing embryo whether it's developing into part of the head or part of the thorax, whether it should be producing legs or wings, or whether it's part of the abdomen. So Hox genes were first discovered within Drosophila, this model organism, the fly, and it was noted that these genes are expressed in a particular sequence down the embryo as it develops, and that they're also, these same genes are also in that same sequence on the chromosome or chromosomes on which they occur. So there's a congruence between the physical order on the chromosome and the order in which those genes are expressed down the developing embryo. Now, when these were discovered, it was thought perhaps that they might be, well, they were certainly known to be a patterning mechanism in flies. They were thought maybe they might apply across insects, maybe across all arthropods, so insects and spiders and crustaceans. Maybe that was the patterning mechanism that went a bit deeper. What wasn't realised was that they're pretty much ubiquitous across all animal groups. So we find them as well in mammals, let's say. And we find them indeed all the way back down the evolutionary tree, right the way down to things like jellyfishes. It's simply that the number of copies of these genes has increased in the more complex groups. So homologous Hox genes within mice and within insects. So again, spanning a huge tract of the evolutionary tree and a hugely deep divergence within evolution. So again, a bit of evidence for common ancestry. Now, all of these homologies are internested. So there are lots of things, for example, that you share with a chimpanzee. There are maybe a fewer things that you share with a cat, and so on. So the internested pattern of homologies tends to suggest a tree, and indeed we use those homologies, both morphology and molecules, to work out the structure of the evolutionary tree of life, and that's something we'll come back to in a later lecture. Another bit of evidence for evolution is something called convergent evolution. Let me give you an example of this. This animal on the left is a fish, or rather it's the eye of a fish. The animal on the right, the eye on the right, belongs to a squid. So this is a fish, so it's a vertebrate. So it's a distant relative of you. This is a mollusk. So squids are mollusks, more closely related to snails and slugs than it is to you. So if we plot a very simple back of an envelope phylogeny of the major groups of animals, then the fish sits down here within the vertebrates, and the squid sits up here within the mollusks. About as far apart different ends of the evolutionary tree as you could hope to be. And if we look back into this tree, there are lots of things deep within this tree that not only don't have eyes of this type, but they don't have eyes at all. Now, the similarity between the eyes of squid and the eyes of fish is quite staggering. They both have a lens, they both have a cornea, they both have a retina, they both have an eyeball, they both have an ability to focus images at different distances. So there are lots of very detailed similarities between squid eyes and fish eyes or other vertebrate eyes. But, and this is the thing, they have evolved independently. And we know that they've evolved independently because of all the other aspects of biology, morphology, and molecules that allow us to place squid within mollusks and fishes within vertebrates. So convergent evolution is a phenomenon whereby animals and plants come up with similar solutions to similar problems. Sometimes at different times, they evolve at different times, and sometimes in completely different branches of the tree of life. Here's another lovely example. Now think back to the slide I showed you with the platypus and the two groups of mammals, the placental mammals and the marsupials. Marsupials and placentals, as I say, diverge way back in the early part of the Cretaceous, and they evolve on different continents by and large. So they're two independent experiments in what it means to be a mammal. The animal on the bottom is a dog or a wolf. The one on the top is a thylacine. So the one on the bottom is a placental, the one on the top is a marsupial. The similarity between those two skulls is quite stunning, even in terms of the teeth and the shape of the zygomatic arch, the cheekbone, the presence of this crest on the top of the skull for muscle attachment, both in the dog and within the thylacine. So this is more closely related to a modern wombat or a kangaroo. This is more closely related to you. So convergent evolution. This is the thylacine wolf or the Tasmanian tiger. Sadly now it's extinct. The last one died, I would say it died in captivity in Hobart Zoo in 1936. That's not actually true. Unfortunately, it got locked out of its enclosure and died of exposure, rather sadly. Nobody knew that this was the last specimen, and I'll post up a little bit of footage of that last thylacine. Terribly sad. No longer with us. Only stuffed specimens that you can see now. This is another example of convergence within placentals and within marsupials. The thing on the right is the saber-toothed tiger, Smilodon. The thing on the left is a thing called Thylacosmilus, and this is the marsupial equivalent. It looks a bit like a Smilodon designed by a committee, I'm sorry to say, but it's converged in the morphology of these saber teeth amongst a few other characters. Not quite as nice as the thylacine wolf, but there we are. That's a reconstruction of what these two things might have looked like. Here's another example. The thing on the top you may recognise as a woodlouse. This is a crustacean. The thing on the bottom is a glomerid millipede. So the crustacean, the isopod, the woodlouse is more closely related to a crab. The thing on the bottom is more closely related to centipedes. Well, it is a millipede, so more closely related to this Scutigoromorph centipede over here. They're both arthropods, so they're both animals with jointed limbs, but these are two rather different branches of arthropod evolution. Again, these two branches would have diverged maybe 600 million years ago. Here's another rather silly example. The thing on the left is a hummingbird. The animal on the right is a hummingbird hawk moth. Even drinking from the same sort of flowers, the hummingbird has this lovely elongated beak. The hummingbird hawk moth has this elongated proboscis made up of mouth parts. It's quite likely in this case that the hawk moth is mimicking Batesian mimicry of the hummingbird, which provides it a bit of extra cover, as it were. But again, this is converged. This one is an insect, another is a bird, a vertebrate. Again, converged on a similar form and mode of life. Thirdly, vestigial structures. So the classic story about your appendix is that it has no function. It's a bit of anatomy that's left behind. In your distant ancestors, the appendix was probably housed commensal bacteria, may have been important for digesting cellulose and other things. We have a tiny appendix now, and from time to time it gets infected and then it becomes life-threatening. You have to have it removed. So potentially it can kill you, and if we didn't have clever surgeons, then it would kill quite a number of people, I'm sorry to say. Thankfully, we do have clever surgeons, but it may have a function, and this may be true of lots of structures that we think are vestigial. They may have functions that we just don't know about. So one story says that the appendix is useful for harbouring our natural healthy gut flora at times when we have infections. So the infection sweeps through the main part of the gut and you have a horribly bad stomach and everything just sort of comes out, and your good bacteria, your natural gut flora, is stuck in this little refuge here within the appendix, and it survives and can then recolonize after everything's gone through the system. So maybe it has a useful function after all. But there are structures that clearly don't have any useful function. Some whales, although they don't have any hind limbs, still develop a pelvis, the remains of the pelvis. They just sort of float around, and they don't even join up to the spinal column. But in the embryology of whales, in the development of whales as well, there are structures that are vestigial. So, for example, most of them go through a phase where they have hair, but then that's lost in the adult. Many of them start with the nostrils at the tip of the skull, and they gradually migrate backwards into the position of the blowhole on the top of the head. So it's a vestigial or ancestral position for the nostrils, and then they gradually assume the new position, sort of recapitulating or running through evolutionary history within their development. If you concentrate very hard, you can waggle your ears, or many people find that they can. Hugely frustrating thing to try to do. If you want to waste some time, it's a great way of doing it. But of course, many animals are able to move the pinna of the ear in able to better locate sounds, and we don't really do that anymore. So the muscles that move the ear are vestigial. We've still got them, we just haven't done away with them, but they don't seem to have any effect on our ability to do what we do. And here's one last example. This is a mole called Spalax, and it, well, as you know, moles tend to live a lot of their time underground where it's dark. Spalax has eyes, but the eyelids are sealed shut. One problem with eyes, if you're tunnelling, is that they tend to get grit in them, they can get infected, and that's not very good. Spalax, presumably the selective advantage of having eyes, functioning eyes, were outweighed by the disadvantages of them getting scratched and infected, and so it sealed the eyes shut. So the eyeball is still there. It probably has a function in sensing light intensity and so on, but the structures that would allow that eye to form an image are still there, and the eye clearly doesn't form an image anymore. So it's a vestigial, those structures are now vestigial. Okay, I'm going to pause it there, and part 2 of the lecture will be below in Moodle.